Welcome back from break, everyone. Our next session is under the title of The Landscape of Change, Disruption and Transformation Across the Cancer Care Ecosystem. This panel actually will touch on the revolution happening in personalized medicine and treatments. A reminder again to everyone, please feel free to put your questions in the question box. There will be time for Q&A at the end of this, and we welcome any and all questions from our audience. So let me move now to introduce our panel. First, I'd like to introduce Richard, who is the CEO of Exactis Innovation and a visionary executive with 27 years of pharmaceutical experience. Richard has been a chairman of the board of CQ2M since 2016. Dr. Jack Kitts was recently named CEO of CPIN, the Canadian Personalized Healthcare Innovation Network, following many years as president and CEO of the Ottawa Hospital, where he retired from in June of 2020. As many of you likely know, the Ottawa Hospital is recognized as one of the largest and most important research and teaching hospitals in Canada. Jim Woodgett was appointed as the president and scientific director of the Terry Fox Research Institute in August 2021. Between 2015 and 2021, he was director of research at the Lunenfeld Tannenbaum Research Institute at Mount Sinai Hospital, where he currently maintains a research lab. Today, we're very privileged to have Dr. Aaron Schimmer, Research Director, Senior Scientist, Staff Physician at Princess Margaret Cancer Centre, University Health Network, as our panel facilitator. Dr. Schimmer is also a professor in the departments of medicine, medical biophysics, and Institute of Medical Sciences at the University of Toronto. He is the past president of the Canadian Hematology Society. Aaron, I pass it over to you for a great session.
Thanks so much, Anne-Marie. It's great to be here. And I got to say, we've got such a fantastic group of panelists today. I think we're going to have a great discussion talking a little bit about the future of clinical trials, and particularly in this post-COVID era. And, as I think about clinical trials during COVID and my role as both the staff physician and research director, COVID's touched all facets of life, and clinical trials were no exception. That we saw clinical trials put on hold or often even stopped due to COVID. We wanted to minimize patients' visits to hospitals. Monitoring of trials became difficult or at times impossible. And of course, all the negative aspects of COVID, I would say, are well known. But there are also actually some positive things that occurred that maybe don't get as much attention. So, for example, we saw collaboration occur at a speed and scale that I think we've never seen before. Sharing of data, sharing of reagents between academics, between institutions, even between academia and industry, happening in ways we haven't previously appreciated. Agreements that would take 6 months to a year to do could be done overnight. Clinical trials could be reviewed and opened in a week. Now, some of this is because perhaps people were working 24/7 and there was an emergency at stake, but maybe it also highlighted that we had many steps in our process that perhaps weren't needed. But beyond COVID, as we start to move forward, science has progressed too. And I think we're going to see that clinical trials are being different in this post-COVID era. For one, at least from where I sit as a physician and a research director, I think trials are becoming more complex. And complex, I would say, because more of the therapies are more tailored and more personalized and more targeted. And as a result, trials are becoming richer in terms of correlative studies. We're seeing that they're more focused on narrow groups of patients, and so that individual centers probably don't have the critical mass, even large centers like ours at Princess Margaret, to do trials by ourselves, and greater collaboration across centers is going to be needed. And as a result, that increases the cost of trials. And so the bar to demonstrate efficacy goes up, and the expectations around success in a phase 1 trial, in my opinion, go higher. So we're entering a new era, which I think is going to present challenges, but it's also very exciting. And with that, let me pass it on to hear from our other panelists, and then we'll get into a bit of a discussion around these topics.
Well, thank you very much, Aaron. I'll begin by speaking to the title of the session. The humanitarian crisis caused by the COVID-19 pandemic absolutely changed the game. We achieved something that no one believed was possible: a new and effective vaccine administered to people in Canada and around the world in less than a year. I understand that drug development typically takes an average of 10 years or more before a drug reaches the market. COVID vaccines were not only developed in an accelerated time frame, this was accomplished without compromising ethics or regulations. Two questions arise from this incredible accomplishment. How did we do this? And how can we do it again? I'll speak first to the former question: how did we do this? Key to the unprecedented speed of vaccine to market were three factors that coalesced in response to the gravity of the crisis. The pandemic united the world with one shared vision: to develop, approve, and deploy a vaccine as fast as possible. Forces rallied to support this vision, top down by every nation's government, bottom up by citizens of the world, and in the middle by all of those able to contribute to the solution. Leaders in drug development and approval processes had access to a veritable tsunami of shared global data, and through collaborative efforts, managed to achieve more than anyone thought was possible. Government, industry, and scientists came together on a global stage in an environment of trust, transparency, and collaboration with a common goal: to save lives. To the second question, how can we do this again? This vaccine development was a global effort, and the urgency of time, the shared goal, and the unparalleled access to abundant data cannot be replicated. But what can and should be taken forward are the value and contributions that sharing data can provide. We have data, so the challenge is, how do we access and use it to open the door to innovation in revamping the processes that bring life-changing therapies to patients, as we did with the vaccine? Since beginning my role at CFIN almost 4 months ago, I've learned a great deal about the complexity of developing new drugs and technologies and the processes necessary to bring new products, safe and effective, to be approved. I've also learned a great deal about the work required to create a high-quality, safe, and sustainable healthcare system for all Canadians. I spent almost 40 years in the Canadian healthcare system. I practiced medicine and oversaw a hospital as the digital era unfolded. And I watched as administrative, clinical, and other health and healthcare data accumulated, waiting on the promise that real-world data would one day be harvested and translated from unstructured, siloed data to real-world evidence, providing insights that would lead to better, evidence-based care and outcomes. I also know that real-world data holds great potential to complement clinical trials and bring targeted healthcare solutions to patients faster. This promise and potential has yet to be realized in Canada. Today, most real-world data remains in disparate information systems, left untouched and unused. And although there are still far more questions than answers about how real-world data can be converted into real-world evidence and used in today's healthcare system, what happened with the vaccine is proof of concept that the impossible becomes possible when technology, healthcare data, and determination come together. And because of that, a more robust, dynamic healthcare system enabled by data and innovation to improve health outcomes of all Canadians is on all of our radars. Before us is an incredible opportunity to be different in the post-COVID world. Imagine all health and healthcare data being accessible and an integral part of healthcare. Imagine mapping the data journey with assurances in place so that it is quality and decision grade. Imagine all of this enhancing and accelerating drug development and other healthcare technologies. CFIN's goal is to bring people together to co-create a data ecosystem that leverages real-world data to accelerate innovation and expedite adoption of personalized healthcare solutions. Today, I'm pleased to be here to listen and learn of your challenges and your successes, and to explore how CFIN fits into your world. And with that, I'll transfer to Jim.
Thanks, Jack. So I'm the new guy on the block. I just become the president and scientific director of the Terry Fox Research Institute, which has been well known for its support of researchers, fantastic researchers across Canada in the field of cancer research. So when I joined TFRI, as it's called, I wasn't so familiar with a new program that they've been developing with Health Canada called the Marathon of Hope Cancer Centre Network. And this vision, actually, which is now in place, brings together the leading cancer centres across Canada. This is going from coast to coast to coast, including the Atlantic provinces, Quebec, Ontario, the Prairies, and British Columbia. And the idea behind this network actually is to reduce to practice an area of research which has really come into its own in the past decade. And as Aaron alluded to, this requires us to work together very closely. And actually, he's head of the Princess Margaret Cancer Centre Research Institute, which is the largest cancer research institute in the country. And even that isn't big enough on its own to do the types of clinical trials and the data associated with those clinical trials to be analyzed in a way which we can really delve into the intricacies and details associated with the type of data which we can now recover through genomic profiling, for example, and other types of profiling. So what does that mean? When we talk about precision medicine or precision oncology, it means that there's a transition from dealing with a particular site for a cancer. It may be breast, prostate, colorectal, and recognizing that some of the lesions, some of the drivers which drive the cancer and which are required for its for the growth, the rapid growth of the cancer, can be shared between different cancers independently of their site. And the advantage there, of course, is if you've developed a therapy, a drug, which is tackling and targeting one of those lesions, one of those molecular abnormalities, and it's been proven in a clinical trial in, for example, ovarian cancer, but that same gene or that same recombination event occurs in lung cancer or in colorectal cancer, then that can be applied, that same drug can be applied and often has very good effects. But we don't look at it, we haven't looked at it in the past in that sort of light. We've tended to treat cancers based on their site, and that's changing. But to do that, we have to build together these huge resources, these huge databases, which compile genomic information, outcome information, patient data, patient information in a de-identified manner, and then look for clues as to the types of molecular lesions and the outcomes following treatment to those patients with a particular therapy on a particular trial, for example. And that requires we share data. It requires that we get together and collaborate. And it's not as though oncology researchers don't do that right now. Of course they do. But for many good reasons, we actually have many barriers between our institutions, between our provinces, and of course around the world. And so those are elements which have to be reduced, have to be negotiated and worked around in a productive manner to accelerate the type of research that we want to do. We're entering an era where individual researchers working with individual patients for improving outcomes is not enough. We need to collectively work together, and that's what the Marathon of Hope Cancer Centre Network is trying to do. And in collaboration with that, there's also the Digital Health and Discovery Platform, which is a partnership between the TFRI and Imagia, a artificial intelligence company based in Montreal, as well as at the moment about 35 other partners around the country. And that group is tasked with providing that platform for digitalizing and sharing and exchanging information in a safe safe manner and retaining privacy, but also allowing us to use, for example, big data and machine learning and computer algorithms to look for patterns which we as scientists are not necessarily can identify ourselves. In looking for patterns in the data, which really overwhelm our own ability to understand that. So together, these two programs, the Marathon of Hope Cancer Center Network and the DHTP, we hope to work with oncologists across the country and really bring precision medicine to all Canadians. I think I'll stop there. And over to you, Richard.
Thank you very much, Jim, and pleasure to be following on. You've certainly set the stage, Jack, Jim, and Aaron, to make it very easy for me to move forward and comment a little bit more. Certainly, COVID has taught us many things. An important learning is collaboration, and when we put our minds to it, how rapidly we can accelerate the outputs and impact of research on helping deal with critical disease. For sure, COVID has delayed much of our work, particularly in delaying, and as Aaron has already mentioned, even stopping clinical trials. However, the invention and innovation continue. And so just to start with a simple definition or what precision oncology does. It's been stated many times, including today, but here's a couple of graphs. And the one on the left is where patients are not pre-selected on any biomarker, and some of these innovative drugs are provided. And we see that there's not that much difference between adding the drug to a current therapy in a combination or versus looking for something like the PD-L1 expression in the patient population and treating them with a checkpoint inhibitor on that basis. And we see that many more patients in the cohort than in a cohort can benefit. One of the challenges, of course, is that when you look at the long term, resistance seems to occur anyway. And so it underlines how important clinical research is in order to move things more effectively and extend survival and bring it to the bedside. And now on the second slide, over time, we see that roughly 40% of clinical trials are selecting patients or stratifying patients on the basis of a biomarker. And if we look at immune oncology, which is more recent, this has jumped to 20%, where things like PD-L1 are a very important marker to recruit a patient and to administer a particular drug. However, the rate-limiting step, and this was mentioned as well, even Princess Margaret Hospital can't catch a large enough patient population to be able to represent a large enough trial population across different therapeutics because of the rarer nature of these molecular aberrations, not only in the tumor, but potentially also in the patient as well, but mostly in the tumor. And so collaboration becomes the critical factor. And the collaboration is really not just among scientists, but it is among major stakeholders. The public-private contract in R&D is fundamental, and that starts with the patient, and it starts with the oncologist in our case, but it also moves to the cancer center and the clinician scientist and the international community of pharmaceutical companies who are doing much of the drug discovery and clinical development of these drugs. So our ability to pull everyone together and row in the same direction is very important. So the bottom line here is that it takes a network and it takes a dedicated infrastructure and multiple partnerships to execute precision oncology. And so Exactis Innovation, we're not in the same space as the Marathon of Hope Cancer Center and Terry Fox, which are further upstream, and they're looking at much larger data series and use of artificial intelligence and machine learning to start to pull out novel and discover novel trends and patterns and biomarkers that will not only drive developmental aspects, but probably fundamentally much more important in discovery and helping industry and other researchers focus their invention to tackle these new findings. What we do is further downstream with a network of 16 cancer centers, which took us 5 years to build, and principal investigator coordinators that help organize our work and our focus and standardize our methods using five biomarker screening labs, et cetera. Because what we try to do is to match the patient to the trial, and we work with the industry very closely to access their pipeline so that the populations that we try to reach out to, that they make sense to the trials and the pipelines and the medicines that are available. So the coordinated, standardized, and common approaches in IRB, contracting, et cetera, are fundamental to help move the adoption of this kind of research into systems perhaps a little faster. And the other important component, certainly, and coming back to resistance and the appearance of resistance with any single therapy, whether it's precision or not, these challenges remain, and cancers are so adaptable and also respond to intervention, is the fact that combinations are probably critical to move forward and help patients survive longer. And in order to do that, that means that our collaborations and our networks and our interplay among networks and organizations is to try to achieve longitudinal capture of data and monitoring patients across the trajectory of their disease, which would include progression events and making sure that we can capture what has changed molecularly in their cancer. And so what I'd like to underline with these few slides is that one of the major obstacles or bottlenecks that we need to overcome is to find ways to have greater access and broader access at the bedside for cancer patients of molecular testing. And we're not talking about companion diagnostics. We're speaking about panel-based, NGS scale or scope testing, so that we can capture these tumors. As Jim had said, you can have a certain aberration that appears across multiple types of cancers, and there are multiple aberrations. So to be able to capture these in one go requires a large patient catchment, but it also requires the support of the provinces to help us, move and reimburse and get more of the testing going. Three key things about clinical trials, whether there's COVID or not, these things remain, and we need to catch up because we've lost time and more patients have delayed diagnosis. But clearly, as I mentioned, broader access to molecular testing that includes reimbursement, but also the adoption of innovations such within the testing, such as blood-based assays. These would particularly have high value in non-small cell lung cancer, for example. So we need to look at multiple platforms and modes of testing. Immuno-oncology has improved survival and durability, but also demands a composite view of the tumor host status. And that means not only measuring the what the tumor is expressing, but also a snapshot and a longitudinal perspective of what's happening with the patient's immune system in response to the cancer and to the drug treatments. And finally, combinations, clearly a more rational combination is demanded, and trials in order to study these complex pathways that any one patient will follow requires that we start to explore more of the adaptive approach to clinical trials, where molecular testing will guide not only which drug to start with, but which drug to combine, and then at progression, which arm or which therapy options to assign patients in a dynamic and real-time manner. And these are areas that are extremely challenging, but come back again, that no single network or no single site can achieve. It requires all partners and all stakeholders to really work together to move that forward. So I think I'll stop with that at this point in time. I've probably eaten up my 5 minutes. And Aaron, so back to you. Thanks.
Super. Thanks a lot. Look, that was a great overview. I think that we've touched on some exciting points, and over maybe the next 35 minutes or so, we'll have a little bit of a discussion, a bit of a free-ranging discussion. But I want to start with the idea around collaboration, because I think, each of you have touched on that to a greater or lesser degree. And Jack, we'll start with you, and I want to talk about what I call team science. And I think that clinical trials require a team. It's traditionally a team of clinicians at the same or maybe different institutions. Maybe that team includes a pharmaceutical industry partner. But I wonder, Jack, is that team expanding or is it changing? And what your thought is?
Yeah, a very good question. Thank you, Aaron. First of all, as I said, I've been in healthcare a long time, and I've seen us go from very siloed, non-sharing, non-collaborative working in the healthcare system to a lot of collaboration. Both Jim and Richard have showed great collaboration in their companies, collaborating with others to 16 cancer centers and so on. So I want to say that there's lots of collaboration, way more collaboration, but I wonder if it's still too siloed. In other words, looking at improving the time to patients, the approval uptake. Would it be possible to bring everybody engaged in the data journey to map it out and look at whose roles and responsibilities it is to ensure that that the data, when it gets to the decision point, we can reassure that it's the right data. It includes clinical data, quality of life data, economic data, genomic data, and so on. So have we put together a group for that? Do we really trust that the data in real world coming out of health records is really, truly quality data? And do we have a measure for ensuring that there are some standards to certify that it is? And then is it cost effective how we're curating data now? And will artificial intelligence and whatnot help us get a data flow that is trustworthy? And then to me, the final point is, can we federate the data and can we agree to share the data? And I think to do that, it starts with trust. And I think collaboration is a result of creating an environment of trust, transparency, a shared goal, like in COVID, and shared leadership to really take it from time zero. Let's agree on what is the question we're trying to answer with the data, or what is the problem we're trying to solve, and then make sure that the data journey ends up in a point where the decision makers feel confident that they have the right data, the quality of data, and the evidence to make a decision. So I guess what I'm saying is we jump to the processes, but I think we underestimate the importance of spending time creating that trusted, transparent environment. And I'll leave it at that.
Thank you. Jim, I'm going to turn to you. You touched really nicely, I think, on the role that Terry Fox is playing in clinical trials, clinical investigation. And I wonder, does this, you think, represent a philosophic change for organizations like Terry Fox or other granting agencies, where perhaps, the granting agency used to be an organization that used to distribute funds to other, usually academic groups? Do you now see a philosophic change where Terry Fox is more involved in facilitating collaboration or maybe even helping advance scientific direction?
It's a good question, and I think what's happened over the past few years is that it's not as though the Terry Fox Research Institute was only focused on basic science. As you know, we've been supporting a lot of translational research as well, moving things towards the clinic. But it's what I think has been made abundantly clear from the amazing science which has developed over the past 20 years with the completion of the human genome, for example, is that this technology really does have a place and an important role in clinical research and in clinical application. What we struggled over that 20 years is how to close that loop, how to take the technologies which have been difficult to establish in many research centers, how to take that into a more clinical setting under clinically approved guidelines and to make use of it. So, for Terry Fox, it's actually, we're closing a loop there, which was artificially left open. And I think there's been so much development in the basic science of understanding our genomes and our proteomes, which is all the genes and all the proteins which comprise us, and then making use of that and applying that technology and that new information to clinical trials and to clinical practice. And as you and I know, it's all well and good to do something in a research lab, but translating that into clinical practice, this is something that Richard alluded to. You have to bring in all sorts of other elements as well. You have to prove to the payers, in Canada, the ministries of health, that it's worthwhile doing. You have to prove to the pharmaceutical companies that a clinical trial is worth doing and is worth doing in your institution because you're asking the right questions. And that's not always easy because especially in terms of precision medicine, often what we're doing is reducing the number of potential patients because what we're doing is identifying the patients who are actually going to benefit from the therapy and excluding those or helping to those who are not going to benefit to be participating in another type of clinical trial. And the drug companies aren't necessarily too keen on reducing the number of patients they see, but of course there's better outcomes. So it's complicated, and the shorter answer is we should all be interested in the whole ecosystem from basic science discovery right through to health systems implementation, because only by bringing all those things together can we possibly improve the health and quality of life of Canadians.
Super. Thanks, Jim. Richard, I'm going to turn to you because your organization, I think, is unique. And do you see this fitting now in part of that larger expanded team of team science and how, you see your organization helping facilitate collaboration around clinical trials?
Yes, Aaron, thank you. I think it's critical. Whether we talk about silos and whether we as individuals can break silos down or not. The point is that precision medicine is breaking these silos down because it needs it and demands it. The catchment and the critical mass required to make it a little bit more economical to practice this research area, and even more so to bring it to the bedside, demands large numbers of patients, large numbers of clinicians and clinician scientists and allied staff to make precision medicine possible. So for sure. And what's interesting between CFIN and MOHCC, Terry Fox, our organization, and others, there's such strong complementarity of this sunken cost to have developed our infrastructure. We owe it. It's an obligation to the taxpayer, I think, that we find ways to work together, because the fortunate thing is we're not repeating what each are doing, although I think maybe on the surface it might look that way, but it's not. In our case, our organization's further downstream. It's actually at the matching of a patient, doing some genomic work with limited or broader panels, not necessarily whole genome or whole proteome, but really focused panels that relate to actionable mutations that relate to available trials. And so we're further downstream, and that's where we work. When we look further upstream, we see tremendous opportunity for collaboration. And I think, Jack, you touched on it. In this whole area of data and curation of data and finding ways of accessing, sites, data across so many hospital sites where research is being done, where there's a richness of data. All of these components, we all benefit if we come together. So where we've been successful is, I think there's three key areas that we've looked at. We've looked at blood-based, we got a trial going in blood-based non-small lung cancer support testing. to standards of care. We want to see the utility of that. Can it expand the number of patients, for example, who could benefit from precision approach? Because we know, for example, that it's a late stage diagnosed disease. Not all patients can sustain a core biopsy, etc. And so a blood-based NGS approach would be very helpful. We're also looking at combinations, and we're looking at multi-omics, but it's at the level of a trial. We're not looking for new biomarkers. We depend on everybody else to figure that out. So I think that if we play our roles properly, yes, our organizations have ways to start to collaborate. And I would say that Terry Fox has reached out with DHDP across many sites, and we've been having conversations with them. CFIN has been doing the same. So I think it's a very strong signal these days that we're starting to respond to break those old silos or older ways of doing things to respond to getting this to the bedside.
Yeah.
Super. Thanks, Richard. One of the things that struck me in your comments is the discussion around the molecular profiling, perhaps the genomic profiling of tumor cells. And maybe, we can go there just for a minute. Jim, I'm going to turn to you and get your thoughts. If you could summarize where you think molecular profiling of patient tumors is today, but more importantly, where do you think it's going to be 5 years from now?
So, one of the most remarkable scientific developments in the past 15 years is the reduction in cost of sequencing. I mean, the first genomes for the Genome Project cost $1 billion or so, and certainly multi-millions of dollars. And today you can get a sequence for a few $1,000s. It's quite remarkable. But that's not the real benefit. That's not the real goal of doing genomic research, because you have to understand it. And we all have very similar genomes. That's why we're human. But what you have to do is to have tools to identify the needles in the haystack, the molecular changes, the 1 in 3 billion nucleotides which comprise our genomes, which may be mutated and driving a particular cancer. And that requires complex bioinformatics, computational programming, et cetera, et cetera. So where we are now is that we can do this. It's been reduced to most research labs can do whole genome sequencing pretty quickly and get turnaround in a few days. The challenge is how do you take that into the healthcare system and have somebody else pay for it, because it's still a significant amount of money. The other element to this is that I wouldn't say it's unfortunate, but it's typical of science, in that as the technology develops, we can do more and more with it. So there's all sorts of different types of analysis which can be done, and perhaps again, which is what has come up over the past 10 years or so, is the realization that tumors do not behave identically. In the same tumor, you've got multiple clones or multiple portions of cells, tumor cells, which actually have different mutations. And so they're heterogeneous in their composition. And some will respond to a drug and some won't. So as a clinician, as you know, you see tumor shrinkage, that's a good sign. But quite often the drug, the tumor retains some portion which is resistant to that therapy, and then it grows back. So we have to therefore understand what are the different fractions, what are the different parts of a tumor, and to try to compensate for that heterogeneity by trying to either detect them earlier or to use therapies which are combinatorial and affect all of the tumor cells. And that type of technology really hasn't gotten to the point where we can move it into the clinic. There are some very interesting research trials, but this is an example of we're always sort of behind the curve. In the research labs there's work which is going on, which we know we won't be bringing into the clinic for another few years, but at least we can anticipate this. So the genomic profile today is going to look very different in the future. And the last point I'll make is that tumors aren't static. We often take a snapshot when the surgeon excises the tumor, and describe that snapshot. What was the tumor condition then? But that's actually, as you know, not what we treat typically, or what you treat as a physician in terms of the chemotherapy, for example. If the surgeon's done a really good job, there should be very little residual tumor, but there's been probably dissemination of that tumor, and that's what the chemotherapy is all about. But we're not really studying that. It's much more difficult to study the disseminated, very small numbers of cells which may be circulating throughout the body. And so we need to also incorporate that in terms of following patients over time. And that's a reflection, again, of the success to a large degree of oncology, in that we get much greater survival terms, but patients do often succumb to the tumor through metastasis after a few years or maybe 10, 20 years. So that sort of longitudinal tracking is also very important. Sorry for the long answer, but it's exciting to see where things are going from a research perspective. But what we really need to be able to do is to translate that into better outcomes for patients, and that's going to take some time.
For sure, Jim, but I think you highlight what's not just where we are today, but where it's going in 5 years. And so, Richard, I'm going to throw you the controversial question, because what Jim described is amazing, but it's also expensive. And so how do we keep up with the United States, which is moving at a fast pace? How do we do this in a single payer system? And yes, you've got the hot button and controversial question.
I got the hot button. Well, I think this is exactly it. And, I think we're fortunate because we do have federal and provincial agencies that are committed to the biomedical research field and providing grants and helping us build the infrastructure. And we also have, private foundations as well, such as Terry Fox and many of the hospital research foundations. The research gets this going, and I think all of our organizations have proven that we can look at tumors longitudinally, and we can look at more than one omics platform to study them. But to make the impact, as Jim says, this has to be done at scale. And as you're asking me, well, how do we afford this? And unfortunately, there isn't an easy way to afford it. But there are some basics to get started on. And I think, again, it has to come by combining infrastructures. We can't have a duplication or a replication of similar things being done, across the country in provinces, or even within a city at 2 or 3 sites. We have to find ways to break down some of that resistance or hesitancy to work together and combine the resources. That's one way to make it more efficient and cost effective. I think that's basic. But the fact that to make impact is going to require systems biology, and that means multi-omics, and that probably does mean that we have to look at adaptive arms in studies, and we have to look at more complex study designs and so forth to be able to harness all the learnings upstream on identifying these trends and patterns and the complex heterogeneity, molecular heterogeneity of the cancer, and how it changes under selective pressure of treatment or intrinsic factors as well. So, to me, I think oncology, we have to find a way to engage all centers, all leaders in this area towards a total commitment to make the precision oncology more mainstream, but to organize our infrastructures to be able to deliver it. And it's not going to happen where every patient can benefit, but I think we have to show the way as leaders in these organizations that we could start to form the partnerships to demonstrate the utility and the impact of doing it, albeit at a smaller scale. And then from those learnings, and from the impact, hopefully the economics can be highlighted, that it actually makes more sense and saves lives, avoids toxicity, avoids healthcare utilization to deal with ineffective therapies and approaches by sharpening it and focusing it on in populations who could respond, and maybe through those sorts of baby steps and stepping stones, we can then argue for a larger commitment of funds to precision approaches to cancer.
Fantastic. Thanks, Richard. You know, again, what's interesting about your comments, and I think an underlying current in Jim, your comments, is around data. And Jack, I'm going to throw this one to you. You know, this generates massive amounts of data, right? So how do you organize the data, and ultimately, do you or how do you share that? And Jack, your thoughts on this?
Well, I think first of all, I agree with Richard and Jim. You do need to have collaboration. You need to be in an environment of trust and collaboration. We saw it with during COVID. So I guess I go back to the data journey, and you need everybody at the table who has a say in the data journey, or a veto vote when it gets to the end, to agree on again what are the data sets that we need, what is the generative model for the data. Can we demonstrate that the real world data, which is abundant, is it truly quality, and can we actually measure it? Can we be cost effective in how we mine that data? Because if it's not good data and you spend a lot of money and time trying to use it, and then federate the data. And as both Jim and Richard said, we've met recently, and there's not overlap, because we're supporting each other in terms of trying to use artificial intelligence to bring data. And that's the DHDP, Jim, that you have, looking at federating and curating data. And Richard, we have commonality that we can work together, but we're still just a small part of that whole data journey. And so CFIN is looking at various partners, meeting with many to see who has to be at that table to ensure that by the time the data gets to the decision makers, it's decision grade data.
Fantastic. Thank you. And again, as I think about all the genomic profiling that one's doing, I mean, the purpose is we want to actually do something for the patient with all the data. And that ultimately leads us into new treatments, novel therapeutics. And Richard, we're going to come to you on this one. You know, as we are able to profile, as we are able to do more, you might think that we would be, flooded with clinical trial opportunities from industry. But I wonder, do you think that's the case? Is it easier or harder now to attract clinical trials of novel therapies to the Canadian landscape?
Well, yeah,
Yes, I'm always giving you the controversial questions.
Well, it's a controversial. I wouldn't even say it's controversial. I think it's a reality of that R&D is a global endeavor, and Canada doesn't sit alone. There are also other jurisdictions with quality cancer research occurring, and certainly south of the border and in Western Europe. But increasingly, clinical trials are shifting to other countries and other jurisdictions where perhaps exchange rates might be better, or there is less competition for patients and perhaps easier access to patients and these sorts of things. What Canada does offer is tremendous quality of science, translationally and clinically, and also in basic research. This is known and it has been an attractor for years. But there are a lot of factors that we're a little bit behind on. I think one of the things that we experience in terms of attracting clinical trials is that some jurisdictions are spoiled by having more ready access to molecular testing, to NGS testing, so that the pharmaceutical company is not having to carry that burden. In other words, it's already performed. Whether it's foundation medicine and private plan coverage of foundation and similar types of services in the US, in Canada, we don't quite have that. So when we're trying to do trials, we have to find ways of partially or fully subsidizing the profiling to do so. So I think that's one of the factors that is a challenge. Now, we do have initiatives such as what Terry Fox is doing with MOHCC, and we've covered some. And I know that out in Vancouver, there's the ACT program, and there have been other programs, Octane and others, that have tried to do this. But these are research focused, and they're at a scale which isn't as broad as perhaps it needs to be. So I think that's one aspect to it, and then other aspects is the nature of precision medicine is, as someone has well mentioned, it is a smaller number of patients for whom any one drug in development is going to benefit, and that means assembling many of these drugs together across a disease trajectory. That multiplies the cost of development. While at the same time, we every jurisdiction has limits to how much we could spend on drugs. So I think our environment is changing, or the ecosystem is changing in terms of adoption of the innovation through market access and reimbursement, and getting the drug to the patient covered. And we're a single payer system. Those are definite challenges that we have to face, and the cost of doing it. But at the same time, this ecosystem is shifting as well. And so where does the R&D go? And is it gravitating to areas where it might be easier to move these drugs into the market, into access, or are there challenges to be able to do that? For example, you want to do research and carry on with patients where they will benefit with the therapy post trial as well. Are some of those factors getting in the way? Quite possibly. But I think the bottom line really is that the economics of it is very expensive. And if our ecosystem isn't collaborating and we're not coordinated in the infrastructure function in an optimal way to find the patients, to be doing the profiling, to be doing all the basic work that then makes it easy to assign a patient to a trial, I think that's one of the early, easy issues to talk to. We have to find a way that we can combine our efforts and have the characterization of these cancers on a molecular basis ready and performed. I think this will attract trials more readily. And I think we also, there are many great examples provincially and nationally to look at life science strategies. Health Canada's got one, ICED's got one, every province has one. Let's not create these in a vacuum, in a silo. Those life science strategies also have to work together. And my fear, from some of the conversations we've been having in that broader context, my fear is that these life science strategies may not all be aligned together. And we can't have 10 systems. If we're looking at research, it's global, it's international. We want to attract research here to Canada. We have to be on the same page, and it's a single page, same song. Let's pull it all together. Let's have a common life science strategy.
Super. Thanks, Richard. We're into our final handful of minutes. We've covered a range of topics. And, as we close, though, I'd like to have you give some thought as to what is the area that you're really most excited or optimistic about related to whether it's precision medicine or clinical trials that you see in the next 5 to 10 years. And, Jack, we'll start with you and your thoughts. But, again, what are you excited about over the next 5 to 10 years?
Well, I'll go back to a comment I made in my opening remarks that I've watched the digital era in healthcare unfold over the past 20 years. And each year I anticipated living the promise of digital healthcare that would provide evidence-based decisions, high quality of patient care, more sustainable system, less costly. And I would say what I'm really excited about is that I believe in the next 5 years, we will truly see the benefits of the digital healthcare. And that's because of a lot of the comments that Richard and Jim and I made today. Is we're seeing a growing amount of collaboration. Some of it may be forced, but I think overall, looking back at some of the key factors that provided that vaccine in COVID so fast, we need to find a way to trust each other, be transparent, and collaborate on a shared goal and a shared vision. So I think that's going to happen in the next 5 years.
Fantastic. Jim, same question to you. Before we turn it over to the audience for their questions, what are you excited about?
So I think actually it's echoing some of what Jack just said. But we've a lot of the problems in moving the fantastic science which has been done in the past couple of decades into improving patient care have been on goals. We've wanted to ensure there was privacy for the patients, of course, records and everything else, but we can overreact to that. And I think one really important development which has occurred over the past decade, but really it's still got a long way to go, is in the involvement of patients in terms of these policies. And if you ask people who actually are suffering from the disease, what they want to participate in, or do they want to participate in various trials, what they want that data to be used for, they're amazingly more liberal and open to uses of that data if it's going to help others. And we need to. This is why we need to involve those patients in these decision-making processes, because they, if they get involved and they realize what the barriers are, they're sort of like, why on earth aren't you sharing this data? They understand we don't want data breaches, and privacy is very important. But also, there's got to be a balance, right? And so I think sometimes the clinicians and the researchers are a little bit too protective, and that's going to break down. And that's what happened with COVID. When we look at those clinical trials, the things were accelerated because everybody was moving in the same direction. There was an imperative. Why isn't there the same imperative with a patient with a cancer, which if it's not treated is going to kill them within 6 to 12 months or whatever? That is the same level of imperative. And I know as a leading oncologist, Aaron, you know this every day, and you have that passion as well with the patients. But there should be no holds barred, and we need to be knocking down these barriers and not just accepting them, which we have done for the past 50 years or so. So that's where I'm most encouraged by.
Fantastic. And Richard, we're going to give you the final word on this. Your area where you're excited or optimistic over the next little while.
Well, I'm very excited with the fact that, I look at Ontario right now and I see that the public molecular lab, they've moved to reimburse and they're starting to expand that. Quebec is probably going in the same direction, I hear. The more we can get this across the country, the better. And, I'd like to come back a little bit to our ecosystem and the fact that it is now being recognized more and more how important collaboration is within it. The fact that there is complementarity to so much of the sunk infrastructure. This ecosystem doesn't work on its own. It requires public-private players. We're all contributing to it. We need to continue to nurture an ecosystem that truly respects the value of investing in research, whether it's basic translational, clinical, or even RWE and observational type work. All of this knowledge is critical to help the cancer patient. And it's not going to happen if we're only public focused or we're only private sector focused. So, we do need the pharmaceutical, the biopharmaceutical sector. They're inventing the drugs. They're bringing them forward. We need to create the systems that optimizes precision oncology. That's not going to happen in the private sector. This is a healthcare delivery challenge as much as anything else. And I think by working together and breaking some of those barriers, and Jack, I think you put it beautifully, it's an issue of trust. It's an issue of trust amongst ourselves as stakeholders in these different sectors. We need to toss aside these differences and start to focus on what matters. And in that way, I think then we will see more research done. We're going to see a lot more impact on the cancer, and everyone will be optimizing the role they can play. And I think that's where we're moving towards. In terms of where the discovery is moving, where the demands on us are going. And just by nature, all of our organizations have been reaching out a lot in the last 3-4 months to find ways to work together. So I think we're realizing, we need to move forward to bring this to bedside. I think that's what I'm most excited about these days.
Well, thank you. And Jim, Jack, Richard, I want to really thank all of you for participating in this panel. Before we throw it over to our Q&A session, I hope the audience appreciates what a blue ribbon panel that's put together for this. It's just absolutely fascinating. We've talked about collaboration. We've talked about molecular profiling of tumors, new therapies, and even the future. I think each and every one of these could be its own hour in and of itself. We've just sort of scratched the surface, but I've had a lot of fun. I hope the people watching it did. And again, thank you for being part of this.
Thank you very much.
Thank you, Aaron.
Aaron, I think you need to count yourself as a blue ribbon panel leader as well, in addition to the blue ribbon panelists. That was astoundingly insightful. It's amazing how much information can be translated in 1 hour. We're going to now take questions from the audience. There's several questions coming through, and Aaron, I'll direct those either to you if there's no specific person that they've been directed to, either that or otherwise I'll direct them to the question to the person it's been directed to. While we're waiting for questions to come in from the audience, I'll ask one of the questions related to many of the inequities that currently exist across the healthcare system today. The question would be, does precision medicine exacerbate these inequities? Is there solutions that need to be looked at now to limit the potentially negative impact of the advancement of precision medicine related to some of these inequities? Jim, may we give that question to you?
Sure, I'll have a crack at it. I think actually, yes, that is a really important concern, because as you've heard from today, the types of technologies which are being used are very sophisticated. They're only present in a few labs, or a few tens of labs across the country, and in a few hospitals. So what we have to ensure is that the precision oncology, because of its tremendous potential benefits, is not limited to those in, for example, the major conurbations, and that we can, and therefore further increase the health disparities that we are all very aware of. And one way to do that is to ensure that, number one, that we include multiple populations in the studies, in the assessments, for example, in the collection of the big data, to ensure they're represented. And secondly, we have to make sure that this can be cost effective. And not only that, can be translated into more remote communities through healthcare clinics, through, for example, types of diagnostic testing which don't require whole genome analysis, but only focus on the specific genetic lesions which we've identified as being important for precision oncology diagnostics. So, I think absolutely that's very important. In fact, in part of the Marathon of Hope Cancer Center Network program, we have a specific responsibility to reach out to remote and indigenous communities who at the moment do suffer the highest level of health disparities, because a solution for only one sector of the Canadian community is not a solution that for the future. We need to do much better in equality of healthcare.
Thank you very much. We have had a number of new live questions added as well, and I'm going to ask this broadly to the panel, and maybe Aaron, you'll help me in directing it to either someone specifically, or we do have some time for all of them everyone to weigh on this. It's a bit of a controversial question, which is why I thought it was fair game to ask it. So the question is phrased as follows: the COVID-19 pandemic has highlighted the value of up-to-date information and timely access to health-related data. How do we prevent data hoarding? And so the concept of data hoarding came up in Greg Simon's talk today, very much related to siloed data. Why make it public when you can sell it? In other words, should there be incentives to data sharing or should it be mandatory through federal, provincial, regulatory interventions?
Mute.
Anybody want to take
That's
the
an interesting
question?
question. Maybe we'll see if Jack, would you like to comment a little bit on that, perhaps?
Thanks,
Sure.
Jack.
I may start with a question. Who owns the data? And I think that's where the question begins. Is a lot of public funding, private funding, other goes into the data. So I think the answer to that question is it really hoarding, or is it a misunderstanding as to who owns it and who gets to decide who uses it? And then from there, there's a lot of concern about the data custodian. The data custodians are, frankly, very nervous about sharing data because once it leaves your area, it multiplies in terms of it either a cyber breach or a breach in privacy, and all of that is bad. So I think we have to step back and look at who owns it, who gets to decide to use it, who gets to decide who uses it, and can we share it? And I think both Jim and Richard spoke to the necessity that we need to bring patients, providers, and others into the conversation about who should get to use the data, and who decides when it can be used. So I don't think I really answered your question, but it's an interesting debate. I'll pass it over to one of my colleagues.
So maybe
Any other
phrase
takers?
a little bit differently, the collection and storage of data can be expensive. And if groups are going to contribute to the storage, the collection of data, should they have an opportunity to have an early access? I would say once data is published, that is part of an open community. But Jim, if one contributes to it, do you get an early look?
Yeah, I think this is a changing paradigm, and I think scientists, for some good reasons, have tended to want to keep the wraps on their data until they're sure about what the results show. But I think that's starting to come under question. It's delaying the access to others who may have very different questions and can use that same data to find quite different and complementary types of results. I think as well, if you ask patients if they can, when they consent in signing their consent form, do you want this? Are you allowing your research to be used for public good, or for commercial or for commercialization or commercial use? Most people actually sign saying, yeah, I'm willing to allow it to be used commercially, because a lot of people know that, in fact, it's incredibly expensive to develop drugs, and that's not what we do in academia. We can't afford to do that. We have to work with the drug companies and the pharmaceutical companies to do that. So, I think it's about an educational process. I think the question really comes back to, as Jack was saying, about who owns the data. And it's a really tricky. Data actually isn't owned by the person. It's typically owned by the institution that has been given the wherewithal to actually collect and store that information. But they've got a responsibility not to breach confidentiality and give information out, private health information. So it's a little bit of a muddy field, but ultimately, it's the consent at the REB stage, sorry, not at the REB, at the consent form stage, where that's where the patient gets to make a decision as to whether or not who gets to use that information and for what. But as a patient, you don't tend to benefit financially if there is a commercial discovery made. Those benefits go typically to the institution and to the pharmaceutical company, for example.
Yes, I'm going to make a shameless plug for a session we're having tomorrow, which features some blockchain information, and it really there's a lot of information that shows that putting the patient in control of their data, quote unquote, allows them to sell their data to whomever they wish through blockchain. So tune in for that one tomorrow. Richard, I'm going to go to you with another question that has come through, and it's something you touched on in your closing remarks. Molecular testing is still not standard of care in Canada. How important is it to have standardized guidelines for molecular testing across provinces in Canada? You highlighted what was happening in Ontario, so perhaps I could give that question to you.
Well, I could start it off, and we have expertise on the panel to really color it properly. But I think the most important thing about standardization is that you do want to make sure there's consistency lab to lab, clinic to clinic, and across the country. Whether this is for clinical trials, where it's very important that if you're identifying certain mutations to match to a trial, that mutation is actually there. So certified laboratories and standardized methodologies are critical, in particular at a screening level where you might be using more than one laboratory to find the patients. Equally and critical at bedside as well. So whether we're using a DNA-based NGS or we're looking for using other omics platforms, it is a critical step to organize and standardize methodologies for patients and for clinicians and for the work that's to be done.
Great. Any other comments or anything to share there? Otherwise, I'll bridge to our final question. And again, it's I'll let whoever feels best desirous of answering this is, what can be done to speed up faster access to precision medicines for patients without sacrificing safety?
Maybe Jim, we'll let you handle this one. This is another good controversial one. We had the last controversial question to Richard, so we'll let you see what you think about this.
They're tough questions today, which is good. Thank God we've got all of you.
Is that Jim or Jack, Aaron?
Oh, I'm going to say Jim. Yeah, that's right.
I mean, I can start. How do we accelerate? I think in healthcare, it's you can't make mistakes where patients are concerned, and clinical trials are incredibly safe, fortunately, given the standards which we apply, and so there are some limits to how quickly we can do certain things. But there's also bureaucracy associated with it, as we all know, and we do need to do better there. I think we also need to try to increase the numbers of different organizations, different healthcare organizations, hospitals, clinics, etc., participating in trials, because that helps in terms of the pace of accrual of patients, which is a limiting factor. And finally, it's how do we get the companies themselves to be working together? And that can be difficult, but when we've got multiple drugs or therapies being used for particular conditions, it can be difficult to get coordination. However, the fortunate aspect of this is that many of these companies do have actually similar types of therapies which they've been developing, molecular precision oncology therapies. And so it's getting a bit easier because they're building up their stable. And so I think those elements are helping to accelerate, but it's never fast enough.
Great. I think we're going to end the session there. Or I'm sorry, is there, Jack, you wanted to comment?
Being the newcomer to the group, I just wonder, the randomized controlled clinical trials is the gold standard because everything is so controlled and you have tremendous confidence in the data that you're going to make the decision on. There's a lot of real world evidence or real world data out there. And I think being the most naive in the group, I'm sure, we need to find a way to tap into real world data to make it quality grade data leads to decision grade evidence. And I think that's the data map or data journey I've been looking at. And I think if we can do that, we may be able to repeat the amount of data that was available in the pandemic for clinical trials without adding this data to the clinical trial data. Just a thought.
Thank you. Any final comments? Thank you again to the Blue Ribbon panel, including the panel leader on this one. And we'll turn it over now to Barry for closing remarks for day one.
Thank you very much.
Thank you, Anne-Marie, and thank you to all of our inspiring speakers and panelists who presented such high-quality information today, including our exceptional and thought-provoking keynote speaker, Greg Simon. As agents of change, the innovations you are bringing to cancer patients can both prolong and save lives, providing more hope than ever before. Revolution in the development of precision medicines and immunotherapies, coupled with comprehensive genomic profiling, is changing the way that we fight cancer and is transforming clinical trials and the delivery of cancer treatments. As we heard from Greg Simon, if we are to gain access to new innovations in real time, it is critical that we change the way we carry out research, modernize the way we conduct trials, as well as our approach to regulatory approval and health technology assessment, while also adopting new reimbursement models so that cancer patients can obtain early access to and benefit from this revolution in oncology. New opportunities to build trust, collaborate, and share data can more rapidly transform the way we approach clinical trials. We must not only learn from our experience in clinical trials during the pandemic, but maintain a sense of urgency to continue to explore new opportunities with patients that will transform cancer research and care in Canada. Thanks to all of you who attended today's conference, our day one of the 5th annual Patient Group Pathways to Cancer Clinical Trials and Real-World Evidence. I hope you enjoyed it as much as I did. I know I wanted to jump into every panel and be a panelist myself. I know that they had such wonderful things to say, and actually we just have to do everything necessary, as Greg says, to bring about that change and do things differently from what we did before. On day 2 of the conference, we look at game-changing new clinical trials initiatives, perspectives on the modernization of clinical trials in Canada, and investment in life sciences. Central to our role is patient centricity, and we will also discuss how patient-centered approach to cancer clinical trials is driving innovation in the oncology ecosystem. I look forward to seeing you all tomorrow, and have a great evening.
Thanks, Barry. You're offline.